Okay, so for today's webinar, we will talk about the single B cell platforms, and I will emphasize on the power of single B cell platform, like how this platform can accelerate the diverse antibody discovery, not only in the conventional antibody but also like the nanobodies. So for today's webinar, there will be three different parts. The first part will be very quick introduction about different like a classic antibody discovery platform or some novel platforms that you may know or may not know. And then for the second part is the single B cell platform, the introduction from our CUSABIO company, and how we use this platform to discover the conventional antibody, like the murine monoclonal antibody. And then the last part, we will focus on the nanobody discovery. And I think this is a pretty new technique that we just come out to our customers, and not only single B, but also the phage display, but whatever, we will focus on the single B cell. Okay, so introduction about different antibody discovery platform. So first, definitely will be the Hybridoma, right? The Hybridoma technology is kind of serve as a key to unlock the door to the monoclonal antibody applications. And you will see that after this one, there were some like a new technology that failed the limitations of the Hybridoma strategy, like for example, the antibody library, including the phage display and some others. And also we have the single B cell screening strategy. From there, we also have the Phage, and also we have some novel instrument like the beaker and 10x genomics strategy. And after that, there were another one, the proteomics. I think it's pretty expensive, yeah. But whatever, altogether, these different platforms, they made great contributions to the monoclonal antibody development. So the idea for all this is to isolate the antigen-specific monoclonal antibodies, and from there we can develop them into the new drugs. So let's go over one by one very quickly. So Hybridoma, so basically it's rely on the immunization of the animals with desired antigens, and followed by the fusion of the specific B lymphocyte with the immortal melanoma cells. Then after the culture, the hybrid cells, they will be generated, right? And then we will have a culture and select the antibody secreting the positive clones. So with the positive clones, they will be transferred to the large-scale cultures and to produce the antibodies in large amount. So it's very traditional, Hybridoma technology. With the antibody library, I think the key feature for this one is that you can facilitate the selection of antibody with the prescribed properties. Like you are kind of like coupling the genetic information with the protein phenotypes. So it's kind of like a genotype, phenotype coupling together. So you can make different genetic modifications with the vectors. Then you will see the characteristics of the antibodies changed. So for example, like the phage display that I showed you here, this is a pretty successful platform in antibody discovery. So like if you have a different antibody candidates, they can be fused and integrated into the phage genome. And then after the expression, the antibody can be displayed on the phage, right? The phage surface. You have a library of the candidates, and all these candidates will have the desired characteristics that such as the affinity, selectivity, or stabilities. And with the biopanning, you will select the optimal features that you want, and you will get the optimal after the biopanning. Then the next one is the single B cell screening platform. So for this platform, I know that there were different branches, based on this platform, like the B cell sorting and isolation, And also we have something like the microfluid chamber-based culture, that strategy. And we also have the high-throughput sequencing strategy. So slightly difference between all these three strategies. Like for example, for the B cell sorting and isolation, usually we based on the B cell receptors on the cell membrane surface. So you can use the Phage and some other instrument to select the target single B cells. And this type of the B cells, they will, we extract the gene from the cells, and then we have the recombinant expression. Then we will perform the different verifications method or assays, and then to get the target antibody. And for the microfluid, the chamber-based culture. So for this one, we based on the cell screening antibodies. So the single cells, that's secreting the antibodies, they will be isolated and cultured. So with this type of the cells, we still do the gene amplification and recombinant expression, then followed by the validations to get the ones that we want. And except the previous two, the high-throughput sequencing is pretty straightforward. So for this one, we just directly use the 10x genomic or some other instrument to get the single cell antibody sequence. So we directly sequence the single cells and perform the antigen and antibody, some like the protein structure prediction and analysis to get the target antibodies. So, but whatever. So for this platform, the basic idea is that you get the single cells, and then you do the sequencing, gene amplification, and recombinant expression, and then get the target. For the proteomics approach, for this one, it's kind of like a slightly different, but I think the basic idea is the same. So you can have the serine or plasma from the mice or immunized the mice, and then you purify it to get a library. So for this library, you can think it's a reference database, right? And then you will also get meanwhile, you will also have the functional different polyclonal antibodies that from the immunized mice, and then you use the mass spec to get the information. Then based on the reference database and also the mass spec result, the sequence can be identified and synthesized. And after cloning and expression, you will get the target one. So for this proteomics approach is expensive, and you largely rely on the mass spec technique. Summary here, or I will say is a kind of like an advantage of the single B cell technology comparing to all the other three. So you see that when we compare the single B cell with the hybridoma, we can see that the development cycle can be short, and the single B cells can be processed in high-throughput manner, so the efficiency will be high. And comparing to the antibody display, the main feature for the single B is that you kind of like keep the nature pairing between the heavy chain and light chain. And also the diversity is pretty nature. So it has a broad diversity with the single B cell. Comparing to the proteomics, the advantage I think basically is the cost, because I know that the proteomics approach, the cost can be pretty high, and also because the instrument, so the manipulation can be time consuming. So, but single B cells can reduce the cost, and also the accuracy is also high. Okay, so then let's go to the main part today. So we will talk about the features of the single B cell platform from CUSABIO. And in the second part, I will show you two different cases that how can we use our single B cell technology to discover the murine monoclonal antibody candidates. So the feature of our CUSABIO platform is that based on the Phage screening approach, we perform a 2-round screening. So I know that in the market, usually we target the company. They usually either target the memory B cells or plasma cells. But in CUSABIO, we target both the memory B cells and the plasma cells. So we then, with the 2-round screening, we will start the antigen positive B cells. So the idea behind this 2-round sorting is because from the memory B cells to the plasma cells. So during the transition stage, there's a very short window there. And during this short differential process, we use the cell surface markers to capture the antigen positive B cells. And by doing this, we will increase the success rate of the positive chromes and also increase the screening efficiency. So the idea is the 2-round screening. And then the second feature of our CUSABIO platform is that we have this method called the linear expression vector construction. So this is a comparison between our strategy to the traditional vector construction. As you see, after the gene expression and the plasmid digestion, you usually get them together, right? And then in the traditional way, you will do the cloning and then you scale up and then you extract the plasmid again, then do the co-transfection. But in CUSABIO, we use the linear expression vectors. So as you see, after the ligation, since our linear expression vectors already have the operate promoters, terminators, and some other regulatory element, so we can directly go to the co-transfection. So it's kind of like directly transfected into the mammalian cells to generate the recombinant antibodies for the further testing. So as the illustration here, we save the time. We skip 2 days and then we do the co-transfection. So the efficiency is increased. And then also by doing this, we lower down the cost. As you see, if you need to construct different plasmids, you can see that as many plasmid as you prepare, the cost will increase a lot. So with our strategy here, we not only increase the efficiency, but also we lower down the cost. So with the 2 features that I mentioned in the CUSABIO single B platform, this is our workflow about the antibody discovery using the single B cell platform. So the take-home point here is that the functional antibodies in the small-scale supernatant, like the 2-mel, they will be available in as little as 8 days. And if our customers, they want to have the milligram scale, the milligram scale purified antibodies, we can deliver the purified antibodies to them within 22 days. So the whole workflow is here, the day 1, we correct the mouse spinning size, and then we do the Phage screening. Then from day 2 to day 4, we do the gene amplification and the cassette construction. Then day 4, we already started co-transfection. So then by that, the day 8, we already get the culture supernatant. So we can use the small amount of the supernatant to do the functional analysis and the validation. Then after that, we got our functional antibody candidate. Then we will need another 14 days to get the large amount of purified antibodies. So this is the whole workflow here. So then you see that comparing to the Hybridoma, the process can be pretty short. It's not even 1 month, just 22 days, you will get large-scale purified antibodies. So as I said before, for this part, I will show you 2 different cases. The first case is about a target called the TROP2. So this is TROP2 is pretty hot and and a promising target in the cancer treatment. So we already finished this project, and the whole process is that we do the mice immunization, then we use the Phage to screen the antigen-specific B cells, and then after that, we do the linear expression and vector construction, then we do the functional analysis after the antibody recombination. I would say that for this project, I'm not going to show you a lot of very detail, but I would say after the Phage screening, we got 24 leads. So we got 24 leads, and from there, we select the top 3 to do the recombinant expression and the functional validation. So here's the verification result. So the top 3 were selected based on the Phage ranking result. So, we do the ELISA verification, and we also do the Phage. And after the Phage validation, we choose the top 3 to express and validate it again with the ELISA and the Phage. So here's the result. So BMK is the benchmark control. So you can think the benchmark antibody control as a positive control. So you see that we have the 3 candidates that we selected. They have a very good performance, as good as the control. And for the Phage binding result here, we even found that we have two hits that showed even better performance than the benchmark control. So meaning that we are able to deliver the good candidate to our customers, and they have a comparable performance with the positive control. So, and I know that our customers not only care about the affinity, but may also care about the diversity of the antibody, right? So we know that. So here, we use the CDR3 to access the diversity. So we know that the CDR region, they have the heavy chain and the light chain, right? And usually we have CDR1, 2, and 3. And CDR3, we can use the CDR3 sequence to show you the degree of the variability and the diversity. So you can think in that is kind of like a marker that you can access the diversity. So we did that. We extract the CDR3 and sex diversity. So usually based on the literature, people will say that if the CDR3 region, the sequence identity was 80% or higher, the antibodies will be clustered together. So we assume that if the diversity was less than 80%, then we will say the candidate are pretty diverse. So as you see here, the heavy and the light chain, the identity was 47% and the 66% for respectivity. So overall, our diversity is pretty good. So we not only guarantee the good performance of our candidate, but also we guarantee the antibody diversities. So then let's go to another case. The case 2 here is about heterodimer. So this heterodimer consists of the alpha V and the beta 6. So the target is ITGAV and ITGB6. So let's just simply say alpha V and beta 6. This heterodimer is a receptor for the fibronectin. You see that after it recognizes the sequence, the RGD sequence in the ligand, there will be an internalization happens here. So, and because this internalization, it can promote the cancer cells invasion. So its hallmark is that after this, it will activate the transforming growth factor beta 1 to modulate some innate immune response. So here were some scientific idea based on this target. I'm not going to take the details about this, but as you showed, this heterodimer is a promising target that recently. So for this case, the different thing about this case is that we use the humanized mouse, the C38. So we use this humanized mouse to affect the screening. And also another different thing is that we got a total of the 56 single cells. So this 56 single cells was sorted using the cynomolars, the antigen, and also the human antigen. This is because the requirement from our customers. So they want to have a candidate has cross-reactivities between these two species. So we used both of the the species antigens to make the FACS sorting. And we got 56 single cells total, and 40 and 16 cells were obtained for each species. And of this 56 cells, we finally get 27 cells that we amplified the gene. We did the gene amplification. And finally, we got the 24 linear vectors. So the positive rate right now is 88%. And after recombinant antibody expression, we also do the ELISA and the FACS detection to confirm the functionality of the candidate. So in total, among the 24 small scale the supernatant, 8 were positive by ELISA and 6 were positive by the FACS. I'm not going to show you all the data here, but I want to focus on this B160. So this B160 was the good candidate, has is not only react with the human antigen, but also react with the monkey mutant antigen. So as you see, the H15H3, this one is the benchmark control, so which means it's a positive antibody control. And B160 is our hit, the promising candidate. So we did the comparison between the positive control with our candidate. You see that for the ELISA binding result, our candidate shows a very good, better performance than the control. And also when the ELISA binding happens between the target antigen, the monkey antigen, and to the candidate, it also shows a very good performance. So this is the ELISA binding result. We also have the FACS binding result with the endogenous cell line, the CAPN2 cells. So H15, H3 is the positive control, and the B160 is our hit. So this one's the B160 still shows a very good performance and is comparable to the positive control. Okay, so hopefully after I show you these two cases, you saw the power of single B cell platforms in the murine antibody discovery and then the summary slide here is that to emphasize on the feature of our CUSABIO platform again. So we did 2 rounds biomarker sorting process to make sure that we can increase the efficiency to find the antigen positive B cells, and also because our strategy in the linear vector constructions, it short the construction time and also increase the efficiency of our platform, the time and also the time. And right now we have already completed over 10 projects, and the delivery rate was 100%. So you can see that we have already completed different targets, and then all this has been delivered to our customers. Okay, so the third part is another main part here because it's related to our new technology about the nanobody discovery. Before I show you the case that we have, I want to show you some basic knowledge about the nanobody because you may not know that what are nanobodies, right? So the properties of the conventional antibody is that you see the conventional antibody, they usually have a 2 heavy chain and then 2 light chain, right? So then you have two identical binding sites. But from the conventional antibody, we usually can get the single chain variable fragment. And this one has been used in the CAR-T therapy a lot. And for the nanobodies, it's slightly different. You see that comparing to the conventional antibody, you do not have the light chain. And so it enables us to isolate the VHH genetic fragment. And this VHH only have one single antigen binding site. So it's pretty small. And this is a summary table between the conventional antibody and the nanobody. First is the size. I will emphasize the size. So the size for the nanobody usually just pretty small. It's 15 kilodalton. But for the conventional, it can be 150 kilodalton. So because the small size, so the tissue penetration for the nanobody is pretty good. The nanobody, they are able to penetrate the blood-brain barrier, so it can reach some inaccessible area that the conventional antibodies cannot reach, such like the extracellular matrix. And also the nanobodies, because it's small, so the half-life is short, which means that the nanobody can be rapidly cleared from the human body. So, but we still have some strategies to increase the half-life. And conventional antibody have some good longer half-life comparing to the nanobodies. So here is just a summary table about features to give you like another idea about what is the difference. So the nanobody discovery, recently they are developing so fast, but I think it's kind of like can be just falling into similar to the single being, because as you see, so just the animal is different, right? We can use the immunized camelid to get the specific plasma B cells, and then we can sequence the B cells and clone into the display library, and use the different display method, and also as well as the FACS screening to get the target antigen. Or even you can have another library come from the synthetic library. So it's not come from the immunized camelid, it's just from the artificial or single-cell library. But the strategy is similar. Like you can use the FACS screening, you can also use the display method. So all this is try to identify the heavy chain antibody fraction from the nanobody and then use for the following steps. The application of nanobody is well established. So recently here we see that they have been used in different therapeutic and diagnostic development. So see cancer therapy, infection disease, and some even diagnostic imaging. So I would say that nanobody can have a very promising, can give the patient a promising future in the disease development. Okay, so after the basic knowledge, let's go back to the case. So case is very important. This is the case presentation from the CUSABIO nanobody discovery. So in this case, we use the single B cell technology. The target we are talking today is let's just say target X. We will just want to keep it a secret. And if you want to know more, we can talk more. So the target X is an emergent target that showed a very good result for the lung cancer in these 2 years. And here is the overview of the alpaca single B cell screening process. So we do the immunization, then we have the fewer blood samples collected, then we use the FACS to screen the antigen specific B cells. With the high-throughput sequencing and the AI power, we are able to get the good candidate to perform the recombinant expression and the purification. Then the last step will be the validation. So for the target X, we start with the immunization with the alpaca. So we have one alpaca, and we did 4 different immunization at different time points. then we first need to confirm that the immunization is successful. So we did the serum titer testing with ELISA and Phage. So the pre-immune serum samples here, they are the negative control, and we use different secondary antibodies to confirm the titer testing. The anti-alpha, the IgG secondary, this is to assess the total IgG amount. And we also have the secondary antibody called the VHH specific antibody to assess that whether we got enough the nanobody there. And the false immunization result are here. Basically, we will say that after the third immunization and the fourth immunization, the result looks good. And we also confirm it with the Phage assay. It's still the serum samples. We have different secondary antibody group. We have a control here, and the sample group, they can come from the blank control. Pre-immune serum samples, third immunization, and fourth immunization. So as you see, the positive rate. For this group, we are testing the total IgG. So total IgG looks good. And with the anti-alpha IgG 2 and 3, these two are the specific class about the VHH. So we see that the positive rate are still good. We even have a BMK, the benchmark control, the positive control. You see that the positive rate was 100%. So it looks like that after the immunization, we got enough materials to start with. So then the next step is that we will do the cell isolation from the serum blood samples. So in total, we have 3,952 cells. So then we started with the antigen X. With the high-throughput sequencing, and also I mentioned before that we have our own AI platforms. So with these tools, we got 10 hits from this population of the cells, and then we did the recombinant expression, and then to do the next verification. For the validation here, the first is the ELISA assay. So this ELISA experiment is to find the hit, to confirm they are binding with the human target X. So as you see here, we have a very good pool of the candidate. The EC50 was in a very good range, and you see that I highlight this one, the MO2Y27 lead. So why I emphasize on this one is because that we find that this one is not only have it definitely has a very good performance and then comparable to our positive control, but also this Y27 hit, we are surprised to find that this one also cross-react with the monkey target antigen. And in this project, at the very beginning, we are not aimed to select the one cross-react the human and the monkey, but we surprised to find that this one, it has a very good performance. So I will focus on this one. And so this is the ELISA binding experiment between the Y27 with the cynomolgus target X, and you see that we have the control, and the EC50 was comparable to the control. And also this one has a very good performance in the Phage assay validation. So this is the Phage result. For the control, we have the EC50 like this, and the Y27 looks like that is even better than the positive control. So whatever. The Y27 is a very good candidate, and then we are surprised to find that it not only reacts with the human antigen, but also with the monkey antigen. So this is the case that I showed you that how we find the nanobody, the VHH lead with our single B platform. But as I showed you in the introduction, the nanobody can also be discovered with the help of the phage display technology. So we also have this technology, and we also have the case that I will present to you today, how we use the phage display to also find the nanobody. So in this technology, this is the overview of the discovery workflow. For the library, we do the immunization, and then we correct the third immuno blood and the first immuno blood samples to not only to ensure the library diversity, but also increase the antibody positive rate. So then we start from this immuno library, and we use the technology to screening the candidate. So the case that I present here is still about the target X. So meaning that with this target X, we did the parallel two case. One is with the single B cell technology, the other is with the phage display. And with these two, we are both able to get good candidate. So this is the ELISA binding experiment. This candidate, the MO2129 candidate, not only react with the human target X, but also react with the monkey target X. And in the fixed binding result, it also have a very good comparable performance comparable to the bench control. So yeah, looks like that we are pretty good at the single B platform and also the phage display. And then based on these two technology, I showed you that we are able to find the good candidate for the nanobody discovery. Finally is our antibody discovery platform from CUSABIO. So we are able to offer you different target like the antigen. So we can synthesize we can express not only the non-transmembrane protein, but also the transmembrane proteins. And we are also to offer you like no matter the full length antigen, but also like the actual cellular matrix, really based on what you want. And for the species, right now we have the mouse, rat, rabbit, and alpaca. The immunization protocols can also be different based on what you want. We also offer different options for the positive antibody and the stable cell lines. So this is the overall the introduction about our antibody discovery platform. So if you are interested about any project, so you can talk to us. Okay, so finally, please give me several minutes to introduce our company. We are CUSABIO. CUSABIO meaning customer satisfied biological reagent, and we are specialized in transmembrane protein expression and antibody screening. We have 5 different product lines. We have ELISA kit, proteins, antibodies, molecular biology product, and small molecular antigens. Our products has been exported in more than 80 countries, including Europe, America, Japan, and Germany. And also the cooperation between our company and some famous companies are pretty good. So as you see, we have a cooperation with the Pfizer, Sanofi, Zinker, Johnson & Johnson. And thank you so much, and thank you for your time. If you want to stay connected with us, here's the barcode, the linking barcode, and you can scan the barcode and we can have a more communication there. Okay, thank you so much, and thank you for your time.